We are about to jump in, of course, with our session tonight, blood, right off the bat in tonight's channel. My name is Eli Schwartz, and I hope that I have seen you in the past. I see a couple familiar names, so welcome back. Our session, I just want to set expectations, is that we're going to jump in and get a better feel for how the MCAT expects you to understand information and be able to manipulate information about blood in general in the human body. So welcome back. How's it going, Luke? Hey, Anna. Hey, Rosa. Welcome, Christopher. Now, again, as far as our session today goes, I do want to remind you that, of course, the learning goals for today are based off of enabling you to critically think through blood. Absolutely, there's a basic review of blood biology in your review notes. Absolutely, there's more additional information with some of those science videos on your syllabus. But this is hopefully going to make that causal link together for how the MCAT likes to test you and what sort of things could potentially get confused. Let's jump right in. First of all, we're definitely going to run down through blood types, and I'll tell you the most classic way to tackle blood typing questions and the shortcuts that I've found over the years. Additionally, we'll talk about the curves for saturation and, again, general amounts of oxygen for myoglobin versus hemoglobin versus fetal hemoglobin for that matter, and we'll really get a feel for how the MCAT would test you on those curves and what they're essentially representing in case you didn't get a curve, but they wanted you to be able to answer a question about a hypothetical process. Again, we'll talk a little bit about immune system within the blood, and we'll kind of finish out with how blood wants to keep itself in the body and not get out through any sort of lesions or lacerations or cuts, something like that. Now, starting off, though, we do want to talk about blood as a general holistic overview. So remind me, if I'm looking at this separated blood sample that has apparently gone through some sort of spinning apparatus, what am I seeing here on top? This kind of yellowish, clearish stuff on top here. That is absolutely our plasma. That's about the 55% pure liquid plasma material. And the bottom would be all of our cells. So top plasma, which encompasses all of the aqueous mixture of salts and gases and hormones and all that. And the bottom would be all of our, again, cells, our red blood cells, our, again, breakdown of all the erythrocytes. This is our general split. Now, if we did go through some sort of spin, if we did have blood separated, if we were looking at a sample, maybe we would get them separated into two different pieces, but otherwise we would expect it to all be one nice homogeneous mixture of blood. In terms of centrifugation, as of course Tyler mentions, the heavy stuff is at the bottom. So our big stuff like our red blood cells should be at the bottom, and our lighter stuff like our nice aqueous mixture of salt should be at the top. But let's start with that stuff at the top. This big aqueous mixture of stuff. I know that there's some proteins in there. Now tell me down here in chat, give me one protein. There are a couple you could pick from. Give me at least one that you would better find up there if the MCAT asks you a question about plasma proteins. You can say, oh, here's a plasma protein I can bring to mind. Albumin is probably the first one to my mind as well. Absolutely right. Luke, Tadel, Rosa. This is regulating blood osmolarity. This is important for things like carrier for ions or molecules. There could be a couple others you might bring into mind. Boom, Tadel. Immunoglobin. Love it. Role in immune response in general, and we'll talk about that near the last 15 minutes or so of class. The third one that you might think of is fibrinogen. Nice, Mary. Again, this is regulating clots formation, which we'll see at the very end of class. Again, these subsets of proteins are helpful because the MCAT doesn't necessarily give you this particular protein or that particular protein in a sample problem. They might say that a patient is losing plasma proteins through some sort of problem with the glomerular filtration apparatus in the nephron, let's say. Again, we want to be able to quickly think of an example protein. So usually for most people, it's albumin, but you could think of these other ones as well. They should be proteins that are actively available in all plasma. Let's go into a little more detail, though, over those salts. Give me at least one ion form that we should see in our quote-unquote salts in the body in general, throughout the entire flow of the blood. Better be some sodium in there, Tristan, Rosa, David. I want some potassium as well. Now, I like what Tristan put in here, the Na+. I encourage you always to remember the ion charges. Even though this is the bio section, even though we're going to see mostly bio/biochem, this is a sort of crossover area where we might see a question about blood where they're asking us for the redox reaction within the heme subgroup, where we're actually doing essentially redox chemistry. Same thing with blood ions. Yeah, we might get a question all about ions, but they might say, hey, suppose we did electrophoresis on a blood sample, or running a charge through it, where would we expect certain ions to go in terms of the terminals? See what I'm getting at? That crossover in that biochem / full-on gen chem role. We always want to have that on the tip of our head. So again, absolutely sodium plus, absolute potassium plus. I saw calcium 2 plus. If you don't have these ions memorized, jot them down. And of course, the chloride had better be a minus 1. It's the only negative ion that we see consistently throughout blood overall, always. Now, again, there are other ones, other cofactors, other nutrients, other ions that we could potentially see, but these are some good quick ones to have off the top of your head. Let's jump in there with those nutrients I mentioned. Give me at least one nutrient you would expect to find in blood. We're going to get a pretty quick clip here, but this should be some basic information we're pretty comfortable with. Better see some sugars. Glucose is the number one I'd expect to see. David, Tristan, absolutely. What else besides sugars? Amino acids, for sure, Tristan. Vitamins would be another one. In just a kind of general classic case. Now, as far as vitamins are concerned, I sort of put them up here. And fatty acids, Mary, or lipids in general, definitely. Now, as far as vitamins, minerals, on the MCAT, you might see a couple different types of vitamins. You figured out where I'm going here. Absolutely, David. Now, the water-soluble vitamins, there are 8 water-soluble vitamins. So you could, again, memorize all of the water-soluble vitamins versus all of the fat-soluble vitamins, and have them all memorized separately, or you could memorize just the water-soluble ones. Everything else falls under that other category, or you can memorize just the fat-soluble ones. So again, as far as breakdown, as far as classification, as far as memorization, I always encourage you to do the least possible amount of work. Now, as far as water-soluble, if you want to think of it as sort of a general broad category, B and C are the only water-soluble vitamins. You can actually break them down further into the different B complexes, and you get 8 total water-soluble ones. But as far as we're concerned, B and C are the water-soluble vitamins, and the fat-soluble ones would be A, D, E, K. I'm sure that Tyler's going to be recapping that down here in the main chat. But as far as memorization goes, you can quickly say, hey, B complexes and C are the ones I'd expect to see for my water-soluble ones. Again, just always trying to be in the mindset of how to memorize as quickly as possible, how to avoid extra fluff work. Again, memorize one or the other. Now, as far as hormones go, this is a little bit more of a wishy-washy statement. What hormones would we expect to see consistently throughout the entire body? But there are a couple that we would expect to see almost always operating. Insulin is number one, absolutely today. Maybe not number one, but in the top 3 that I would expect to always see operating in the human body. If we have sugars, we should also have some insulin response. Definitely. TSH would maybe be my number one. You got it, David. TSH, the other one I would probably put in the top 3 would be arguably GH, growth hormone. Because I should expect to see that pretty consistently throughout any blood sample. So TSH, of course, stimulating thyroid to produce T3 and T4. GH for growth hormone for blood glucose and musculoskeletal growth. And then my insulin, of course, for our general breakdown of blood glucose and partnering with glucagon. Now, again, we could talk about some other larger overarching functional steroid hormones, absolutely, Tristan, like testosterone or estrogen, which would be always present in males and females. Again, this is sort of a crossover topic, as Tyler points out, between blood in general and the huge hormone and endocrinology regulatory system. I know that Tyler has actually taught a couple of our endocrinology sessions for the channel. So if you have any further questions about endocrinology or about hormone signaling, peptide versus hormone, hit us up over there in the Q&A. I know that Tyler is absolutely available to answer any of those questions. Additionally, though, let's go into a little bit more detail because I haven't yet shown you anything about blood typing. So I have to make some red blood cells, and then we have to talk about what exists on them that is going to give us this blood typing. Remember, all blood cells are produced from the common progenitor cell known as our hematopoietic cell, or I've heard it pronounced hematopoietic cell. The nice thing is the MCAT is not a pronunciation test, so we actually don't have to pronounce these things as well as know them. So that hematopoietic cell is located in red bone marrow, and again, this cell is going to give rise to platelets or immune cells and red blood cells. But for our purposes right now, let's focus on just red blood cells. We'll get to the other 2 in a second. As far as red blood cells are concerned, there are a few basic components to red blood cells that we should be aware of. First of all, their differentiation is in the bone marrow. It's stimulated by erythropoietin produced by the kidneys. Now, the maturation of red blood cells takes about a week. That maturation, the red blood cells are going to lose a couple things, in that maturation process. Give me at least one thing that red blood cells had better lose from their original cell structure that we don't want them to have anymore once they're going to be actual functional red blood cells. Better lose the nucleus to Dell, David, Marie, absolutely. Better lose mitochondria, nicely done. Organelles in general, yes, David, for sure. And the whole point of doing this process, losing the nucleus, losing the organelles, producing hemoglobin for that matter, is, yeah, you're right, Christopher, right, David, to lose most of the stuff inside. We don't want this to be a big ball cell anymore. We want it to be able to be squished flat into a donut-shaped disc. All of that fluff in the middle is gone, thrown out, so we have more room to carry oxygen, but also for other little things, right? Like we don't want them to have mitochondria. We don't want them to be breaking down oxygen, using that for energy source, because then it would be breaking down the oxygen it's carrying around in the first place. So there's some little side issues there with allowing it to do that. Now the lifespan is not a week. Right? It takes a week for it to be made, but the red blood cells are pretty hardy. They should last for about how long? Yeah, exactly, Chris. About 120 days. And I'm giving you these values so you have a decent feel for what we would expect to see on test day. Right? A red blood cell should last for around 4 months, or it should last for around the third of a year. However you want to think of it, these kind of basic values can come up on the MCAT, where they might say red blood cells in a certain individual are lasting about 60 days before being lysed. Now, they're not necessarily going to tell you this is autosomal recessive or autosomal dominant disorder. They're not necessarily going to tell you it's a particular cancer, none of that stuff. But you should be able to recognize some of these values seem off. That doesn't seem like the normal value I would expect for red blood cells. Now, we don't have to know treatments, we don't have to know any other symptoms, but we should be able to recognize common value ranges. So 120 days is common, after which red blood cells are going to be lysed by one particular organ. Help me out, I've forgotten. Maybe, is it pancreas? Is it the lungs? Oh, it's the spleen. Nicely done. Corrected out. Because a lot of people actually think liver. Now, sure, there's a lot of processing power that happens in the liver. Liver is going to uptake bilirubin. There's going to be the iron that comes back through. But the lysing actually happens in the spleen. It is actually one of the few questions that specifically requires us to remember spleen functionality, is red blood cell processing. Remember, the red blood cells are going through that kind of micron sieve in the spleen, where at that 100 to 120 days or so, the spleen has about 3 micron width holes for the red blood cells to go through, but the big donut-shaped discs are about 8 microns wide. So the red blood cells get squeezed down through, pop out the other side, squeeze down through, pop out the other side, etc., etc. But after about 120 days, and we'll talk about the reasons why, red blood cells start to lose that functionality. They can't quite squeeze through, and they might break or snap or get wedged, and then they have to be lysed. So again, they'll be phagocytized, right? The resident macrophages are going to break them down, and the hemoglobin is going to be recycled. Now I mentioned briefly, but I have to say it again, that recycling process, once they're lysed by the spleen, then the hemoglobin is recycled in general by going back down through iron, will be transported through circulation, transferred by transferrin, and taken up by bone marrow again, because we have to have them made in the bone marrow in the first place. That's where the iron has to go. The hemoglobin broken down into bilirubin, though, that bilirubin then goes through circulation, then is uptaken by the liver, and does make bile. So those of you who said bile from a second ago, or liver from a second ago, that's sort of our next step. Once the red blood cells get crushed into pieces and make bilirubin, then that bilirubin gets uptaken by liver and recycled into making bile. So that's the second organ that is important. First one, though, is spleen. Now, as far as this process goes, that 3-micron red pulp, that kind of sieve in the spleen, and these big 8-micron red blood cells squeezing down through, seems like there's an awful lot of flex necessary, an awful lot of squishing necessary. Let's go into a little more detail about how come they can make that squish and what's actually happening here. And then we'll go into the actual little tiny pieces of structure that make that happen. First of all, there's a biconcave shape, and the biconcave shape is important because then we can have things like that cross-sectional area be relatively small. So I do have 8-micron width, but the actual cross-sectional area at any one point is not a full 8-micron diameter sphere. That would be a pretty big problem. If we had an 8-micron diameter sphere, it would not be able to squish down at all. But with that half area actually being only about a 3 micron radial sphere total cross-sectional area, we can actually have it squeezed down through. So we do have that jelly donut shape. Now the biconcave shape is important, but really it can only make that because we have no nuclei, we have no membrane-bound organelles. If we look at this graph here, this figure here on the right-hand side, you already told me a bunch of these things that we have to get rid of. We have to get rid of the mitochondria. We have to get rid of lysosomes. We have to get rid of the SER. The RAR is gone. The nucleus is gone. The chromatin is gone. The nuclear, the Golgi bodies, all of those are gone because we just don't have space for any of that stuff. And if you look at what's left over, we have a little bit of cytoplasm. We've got a little ribosome, and we've got the cell outside. Right? This is about all we need. Now we need a couple other things, right? We still do need to have hemoglobin in there. We have to add in our heme subunits, but as far as red blood cells are concerned, there's really not much left over here, and there shouldn't be. These should not be organized in some sort of overarching, hugely complicated process. This is like our messenger system in the body. This grabs some oxygen over here, messenger delivers it over there, picks up some CO2 on that side, brings it back to this side. Maybe picks up some carbon monoxide by accident. We don't want that, but go through the process of delivering one way or the other way, right? Now, as far as the hemoglobin molecules overall, we have a massive amount of hemoglobins. But let's go into the MCAT questions so far, because we've been going over a lot of the basics of hemoglobin of blood in general, but let's talk about the way the MCAT might ask you something about this at a fundamental level. They might ask, why do red blood cells have to, as an advantageous mechanism, lack mitochondria? Why do they have to do that? Or another way of saying it, how come they can't have it? Why would it be bad for them to have mitochondria? Yeah, because we don't want oxygen to be used. Exactly right, Christopher. I know we kind of went over this a second ago, but this is the way the MCAT would phrase it. It seems like a relatively straightforward question, but we'd have to be very careful looking through the answer choices. So definitely the lack of organelles is going to create more space, but then additionally, mitochondria will produce ATP through something else. OXPHOS, breaking down the oxygen that they're carrying, would make them pretty inefficient. So instead, they better go through some sort of glucose conversion. And again, there's some other reasons behind this, right? Mitochondria would have used up the oxygen. We really don't want them to do that. It'd be like if you had your little sibling carry your cookies up to your room, but you know your little sibling eats cookies. Then you would never charge your little sibling with carrying your cookies up to your room. That would not be a smart move on your part. So take out that factor that could be using up the oxygen. Now, additionally, we should be clear, one of the other reasons that we don't have to have red blood cells with any sort of mitochondria is you got it, Jacob, because they don't really need to go through oxidative phosphorylation. They don't need to be aerobic respiration to produce a whole bunch of ATP. They're fine netting 2 ATP with glucose. Red blood cells do not divide. They do not synthesize their own proteins. Their ATP consumption is very small compared to a lot of the other cell types. Exactly right, David. There's nothing else that you really have to be doing. They're just flowing along with the blood, delivering, receiving, delivering, receiving. Nicely done. And that's a sort of follow-up question that the MCAT could certainly ask you. What about the fact that they're anaerobic? How is their processing going to survive without them having excess of ATP? Well, they really don't have to. They have specialized, so they don't need the extra energy source. Exactly right. And as Tyler is recapping down here in the main chat, remember, there are a lot of specific functional pieces in the human body, and there are also some groups that do lots of multiple purposes and lots of multiple phenomena. So really breaking down which ones are only one purpose, like red blood cells, versus which ones are sort of totipotent and can do anything, is going to be part of actually when we get into immune response. We have to pay attention to which ones are the specific response, which ones are the general response. But we'll get there. That'll be at the end of our session today. Before we do, though, this biconcave shape sounds really awesome. It's great to have this jelly donut, but the jelly donut shape is really only mediated by the fact that we have certain things tied together. Specifically, we have ankyrin and we have band 4.1. So here's that ankyrin here right in the middle, and then our band 4.1 on the right-hand side. And really what's happening here between these is that they're linking proteins through the actin cytoskeleton, the overall shape, through their interaction with spectrin down here in the bottom. Those are the spectrin dimers linked together. Now, this long chain structure is kind of tied together. It's sort of like a spring, sort of like a rubber band, sort of like the shocks in your car. These are structural proteins that are maintaining the integrity of the red blood cell walls, so enabling it to withstand shear forces across the surface, or go through a hole, or move through the rest of the body bumping into things. So when they hit something, they'll spring back into place. Now, this process sounds great. I've got a spring, I've got a slinky, I've got some sort of shock absorbing rubber band in here. It'll snap back into place. But as this process continues over time, what might happen to the springiness or the shock absorbingness of these red blood cells? Of the proteins, really, within the red blood cells. It slowly gets reduced, slowly broken down. I might have some sort of problem with one of the dimers. I might have some stiffness. Exactly right, Tristan. And that's a fine way to remember this. So now that I've got that stiffness, I've got that breaking down, I've got that structure not really maintaining integrity, now we degraded the proteins. Now when I squish down, I can't spring back, or I can't actually squish in the first place. I'm locked in position, and the spleen comes along and lyses me. These are the main important pieces for maintaining the overall structure. But if you look at the diagram we have in front of you, it looks like there's some other stuff that is pretty big and pretty important. Besides these green lines kind of tied together, about 25% of the cell membrane is covered with a particular type of structure. About a quarter of the whole red blood cell membrane structure is covered with a very particular channel. Okay, glycoproteins are absolutely important as well, David, for sure, for regulation, for functionality. But the thing I'm talking about here, before we get into that, is the band 3. Absolutely, Jacob. The thing I want to talk about before we jump into all the rest of the functionality, because we will go into that in a little more detail later, is band 3. Band 3 is an important structural component because, well, first of all, it comprises about a quarter of the entire surface area of the membrane. But beyond that, band 3, just like our sodium-potassium pump, is an ion channel we should be very familiar with by testing. It's an anion exchanger and mediates the exchange of chloride ions for bicarbonate ions. Band 3 also, of course, if we see in the picture, links up with ankyrin and, of course, band 4.1 and links to that cell membrane overall. But this functions to maintain structure, maintains its biconcave shape, maintains it from collapsing or vesiculating, but also is the ion exchange necessary in a blood cell. I was going to explain it, but let me ask you first. Why might it be so important that a red blood cell be able to quickly and actively do an ion exchange? It's a vague question, I'll admit, but it's for a reason. For acidosis, absolutely, David. Acidosis or alkalosis, for that matter, to maintain pH, to maintain balance. Yes, Luke, absolutely, Lauren. And because we could be going into different regions in the body that might be hyperosmotic or hypoosmotic, and the red blood cell has to immediately respond. That's why we have a quarter of the whole surface area available for that sort of fluctuation. Because what would happen if we had a red blood cell that all of a sudden went into a region that was, let's say, hypo compared to the red blood cell? Hypo, of course, low for solutes, had a lot more water in the region that this red blood cell just went into. What would happen if it couldn't pump out all these ions really fast? Yeah, it starts swelling. Exactly, David. Correct. And certainly, Tristan, if that was not compromised by some sort of other, or again, resolved by some sort of other process, it could be compromised. It could lyse. Absolutely. Again, these are those sort of critical thinking questions that go a little bit above and beyond just memorizing the structure. Why is it there? How could it be helpful if we ended up in the wrong sort of environment? That's the way the MCAT likes to come in and test you on this sort of content to make sure that you really are pretty solid on the fundamentals, but also on how it could go wrong. So often what I recommend to all my students is, hey, memorize the structures, memorize the process, memorize the system, and then ask yourself, what would happen if we all of a sudden lost something? What would happen if that was all of a sudden broken? Would we still be functional? Would it be viable? Would there be some other process that's going to come in and actually resolve the issue? That's the thing to be tucking away in the back of your mind, because that's a lot of the way the MCAT tends to test these. Now, going into the actual thing I promised you we would talk about in the first half hour, we have to talk about blood typing. There are a few main pieces within blood typing that are sort of fundamentals that harken back to genetics and pedigree and our understanding of dominance. For example, ABO system is a codominant system. A and B, just to be clear, are codominant over O. So O is the recessive type, and A and B are actually the codominant types. Technically, we could think about this as being codominance AB or recessive O, or even codominance with A and O, but A is the dominant over O. So we would never really talk about an AO system. We would talk about AO genetics and then an A phenotype. And you're right, absolutely, David. O is its own type, if you will, in the system, sort of like my issue I'll have in a second, David, with RH factor. But O by itself just means not having those, does not have A's or B's. So just to be very clear, it's called its own type, but it is not actually a genetic dominant trait or recessive trait for that matter. It's just a lack of A's or B's. To be more clear, maybe we want a picture. Here's A, right? A has these little pink circles. And here's B. It has these little green squares or diamonds. So if I've got A and I've got B, then I have both of those codominant on the surface of my red blood cell. If I have none of those, I call that O. So I hope I didn't confuse anybody by saying that O would be essentially recessive of these, because O is not actually exhibiting anything. Right? O is the lack of exhibiting a protein on that surface structure, or an antigen on that surface structure. Now, the antigen differs in a couple different shapes, right? That would be our sugar modification structure. That's why A's are represented with these circles and B's are represented by these squares, to remind you that it is a modification of that structure overall. And if I have A blood, or an A antigen on the surface of the blood, I had better not be making antibodies to that A blood. Because again, if this is going to be the blood that I make always, then I don't want to be killing that blood. Similarly, though, if anyone new comes in, I do want to kill that new stuff. So if I have A blood, then I want to kill any bees. I don't want any bees in here whatsoever, unless they're bees in real life because they pollinate flowers and they're lovely. But bee blood, kill that stuff. We don't want any of those. So no bees are allowed. So if I'm A blood, I had better not be making any anti-A's. That would kill myself. I would want to, though, make anti-B's. And notice we've drawn these anti-A, anti-B antibodies with the little shapes to grab onto the receptors, because that's essentially what we're looking for here. Recognition of the receptor, recognition of the antigen, in order to make sure that we are killing the correct thing. Now, to Christopher's question, we have to get into a lot more details of chart, which we're going to see in a second. Hold on to that thought. Before we get there, though, I do want to be clear. There are only 3 allelic combinations that could give you a blood type. A dominant A, a dominant B, or a recessive allele set that gives you O blood. Because I saw a couple people confused about what I meant by phenotype versus genotype a second ago. So O blood, just to be clear, would be the total recessive allele set that makes no surface proteins. A is a dominant and B is a dominant, and they can both be codominant and express A and B antigens on the surface. Now, if you are producing these IgM-type antibodies, then there's something we should remember about IgM as opposed to some of the other ones, like IgGs or the others we'll talk about later on. What's the thing we should remember about IgM compared to the others? Specific characteristic of IgM overall. Yeah, absolutely, Mary. IgM I think of as Ig massive, right? IgM, Ig massive, they're big, they're bulky, they're not going to be able to cross over that placenta into fetal circulation. So if there's some sort of ABO mismatch for some reason between the fetus and the mother, not problematic, does not matter. Exactly right, Tadell, right, Mary? Now let's go into what Christopher asked about a second ago, and certainly what Sam was following up with, this massive chart. Let's fill it out here. First of all, we're going to go through AB, and then O as the possible blood types. Now the blood type that would give you A is either all A genes or an A gene with a recessive O. because, again, we'll do this lowercase I for recessive O, for not having it, for not exhibiting. Similarly, for B blood, it could be all B dominance, that's that capital B, or it could be a recessive, not exhibiting O, essentially, as our genotype set. AB can only be AB blood, and O blood can only be two recessive gene sets that give us no phenotype alleles on the surface, no phenotype antigens on the surface. Now, this first column is our basic knowledge we should know, and the second column about genotypes would only come up if you were being asked some sort of pedigree or analysis or genetics question. The real heart of blood typing questions on the MCAT tend to be in the breakdown of antigens versus antibodies and donation to versus received from. And go ahead, raise your hand or give me a thumbs up if this is the stuff you tend to mix up. Donate to versus receive from, or antigen versus antibodies. Because I know this is by far the most common issue that most people have. Thumbs up, 6, 6, 6. Okay, Olivia, Rose, and Lauren, I agree. Let's break it down. First of all, antigen is the thing that actually is generated on the surface. So a blood type had better be generating what antigen on the surface if it's an A blood type. Better be A's. Absolutely right. This is not a trick question. A antigen. B had better have B antigen. AB? Both of them. O? None of them. Again, antigen generating that surface protein. Great way to kind of keep that in mind. Now, antibodies, oh, thank you, Tal. You already recapped it while I was saying it. He's too fast. Antibodies, right? We think of antibodies as the things that are going to kill something. So if you have A blood, who do you want to kill? You want to kill all the bees, right? Oh, don't kill the bees, though, right? Stop the pesticides. But in this case, kill the bees. Anti-B. How about if we're B blood? Who are we going to kill? Kill all the A's. Exactly right, Christopher. Anna, bless, for sure. How about if you're AB blood? Oh, gosh, can't kill A's. Can't kill B's. I guess kill nothing? Exactly right. Absolutely right. Right, bless. We kill nobody. Because, again, we can't make A's, we can't make B antibodies. We would not kill anything. Now, David is going one step further in this, which we'll get to in a second. If we don't have anything in our blood that would kill new people, well, then we could take in A's, we could take in B's, we could take in ABs. We will accept anybody because we have nothing that we can kill. Cool, cool. Now, how about if we're O blood? Well, then we could kill A's, definitely. We could kill B's, too. Yeah, let's kill all of them. Now, think about O blood, then. Sounds like it's not going to be any sort of universal receiver because it's going to kill everything unless it's itself. Maybe it'll be doing something else. You got it, David and Bless. We'll get there in a second. Before we do, let's be clear. A should be able to donate to everything else that likes A. So A blood likes A and AB blood likes A, but a pure B would have killed A's. So it can donate to anything that would accept it. So B blood can be donated to B or ABs. The receiving from part is interesting, though. Remember, everything should be able to receive O blood because O blood has no surface antigens. Since it has no surface antigens, nobody else has anything designed to kill it. If I don't have anything designed to kill it, then everything is going to be happy receiving it. Now let's get to AB. This is really our extreme case. AB should be able to donate to AB by default, but nobody else, right? Exactly, Mary. Right, David? Because A blood by itself would kill B's, or B blood by itself would kill the A's. So it can't donate to anybody else. This is a selfish blood type. It receives from everybody, but it only donates to itself. If you're AB blood, I apologize. Not actually selfish, just your blood is. Now, how about O, though? Flip side here, other extreme end. It can donate to everybody. Because everybody will accept it because nobody kills O. O doesn't have any surface stuff that antibody would recognize. However, it can only receive from itself. So this is one of those reasons, again, why when you hear somebody calling out in one of your favorite medical drama shows, it says, we need 2 boluses of O negative blood stat for this unresponsive patient with no documentation, because definitely O negative blood will work with that person. We will not have hemolysis, we will not have hemagglutination, we will not have a problem. O negative blood will go into everybody. But it's also why if you are O or O negative, you should be donating blood to a blood bank, because otherwise they might not have blood for you, because you can only accept O or O negative blood depending on the type you actually are. Now I keep saying O or O negative or AB or AB positive because I'm actually adding in one more thing. What's that one more thing I keep adding in when I keep adding in that negative or that positive? That is our RH factor, our rhesus factor. Nicely done, Laureen, Rashid, Blast. Now the rhesus factor comes into play. It really is just one more antigen. Just to be clear, it's just one more thing that could be on the surface. Plus means you have it, minus means you don't. It's just like an A or just like a B. The issue, though, with rhesus is that we could have some problems because the antibody to rhesus is not IgM. Let's talk about that problem. First of all, rhesus factor dominant. If you have rhesus plus, right, then you continue to have it in the future. It's either positive, which means has it, or negative, which means doesn't have it. Just like we could think of O as meaning you don't have A's or B's. We could think of a minus as meaning you don't have rhesus. And if they're not IgM for massive, what other Ig types could this be that could cross that placental wall? They're IgG, absolutely right. And the problem with that, as Annalise is pointing out, is we can actually cross into the fetus and we might have some problems. And we won't necessarily have problems all of the cases. This erythroblastosis fetalis is only in a very specific scenario. So let's start with the basic scenario that would be the best for mother and fetus, right? Negative mom, negative baby. Awesome, we're all good. We've got simple dominant inheritance. We've got no problem disconnect between the mother and the child. Even if there's an ABO problem, those can't cross over because they're IgM antibodies. So we're awesome. The antibodies are made. They can't cross in. There's no problem with fetal circulation. Everyone's happy. Now, there could be a problem, though, and that's the heart of a lot of the MCAT questions. Let's make a problem. Let's see what happens. Well, suppose we have a positive child in that maternal issue here. Now, the first pregnancy, they're very clear about this here. No problem for the first one, assuming, of course, there was no sort of complication or no sort of rupturing anywhere in the process. Then the only time that there would ever be any mixing of nutrients or waste or blood flow would be during the birthing process. Exactly right, Ahisa. Correct, David. So then the mother would be exposed to the fetal blood and begin producing some of the Rh antibodies. But otherwise, no problem. Okay, flow over, producing these antibodies, the Y shape, of course, for our producing of antibodies, starting to have a problem. But the first child still circled in green here, still fine. The second child, though, there already are antibodies there. That sounds bad. So now we've got antibodies, and we've got pluses in the inside. Those antibodies are going to go across the wall. They're going to attack the child, exactly right. And then the child now can lead to erythroblastosis fetalis, as we have here on the slide. They're IgG, they can cross over, they're binding to cells causing hemolysis, elevated bilirubin levels, lots of problems. So we don't want that, right? What we could do is what David mentioned a second ago. Throw in some ROGAM. Absolutely right. Now, our ROGAM, of course, is essentially a blocking process, right? ROGAM is an intramuscular injection given to Rh minus mothers that have Rh positive babies, and the injection is about 28 weeks in. At about 28 weeks, again, that 6 to 7 month range, about 90+% of women will have developed antibodies at that point. So ROGAM is composed of IgG anti-Rh antibodies that are going to coat the Rh antigen that is across the placenta, mask it from the mom so that the immune system doesn't know it's there. Again, the whole reason for that, Mary, is because that's when we would start seeing the antibodies being really developed and being aggressive, is about 28 weeks in. Now, certainly administration earlier could be good, but by that point, about 92% of all mothers will have actively developed antibodies that will actively be trying to kill the child. Up until that point, there might be a safe barrier, but as far as the immune response, it usually takes that long to develop a full systemic attack against the fetus. Good question. If we were a physician, we probably wouldn't wait necessarily until we were sure that the fetus was being attacked, but that is sort of our cutoff point. We have to administer it by that 28-week deadline. Again, I hasten to add that's not necessarily a number that you would be asked on the MCAT, but you could be asked about what Tyler was mentioning, that it would take a long time for our immune response against the fetus, and that length of time is the amount of time that we would have as sort of a buffer before we administered RhoGAM. Good question, though. Now, as a follow-up to that, here's the question I promised you that we would see. We absolutely are going to see a question about blood typing and something about hemolysis, hemagglutination, cell death, lysing, whatever it is. I want to give you a chance to try and answer it in the bottom right, but I also want to make sure I give you the shortcut. So I'm torn with this problem. Here's the shortcut. If we're looking for something that caused hemolysis, then that sounds like something real bad. Well, then you can go through the answer choices, cross out any answer choice that has something that everyone would be able to receive, O negative, and cross out any answer choice that might be the same person's blood, B negative in this case. Because we could never have hemolysis with your own blood or with O negative. To be clear, the longer way to do this is to think anything with A's is going to be a problem and go through and cross through, find all the answers that have A's or positives in there. That's the long way to do it. But the short way is to eliminate any answer choice that would be good stuff. We see 95% right off the bat for the correct answer. If you've been in my channel sessions before, you know what that means. You get a BSD. BSD, of course, standing for Black Star of Doom. This is what the MCAT will look like. Look at that. Look at that terrified face when we crush them on testing a question like this. Absolutely answer choice B, because it's got all bad things. A positive or an A is going to cause hemolysis for a patient with B negative. Just to be clear, A had B negative, the blood of the person in the original place, and C had O negative, and of course D had B negative. All things that would be fine for this individual. Nicely done. Now at this point, we do have to transition into the kind of physiology of those curves, and at the very end, in the last maybe 15 minutes that we would have after this section, we'll try and get in as much of the immune system as possible, but immune does come up in some of the other channel sessions, so I really want to make sure we hammer home the curves as much as possible in today's session. First of all, what we see here looks like a big bundle of stuff that only has one binding heme unit in the middle. I know already in the slide we've got hemoglobin, but as far as, myoglobin, but as far as myoglobin goes, it's that single unit, it's found in muscle, it's got a higher affinity for oxygen, and it has that hyperbolic, very sharp binding curve. Now it is non-sigmoidal, exactly right, David, because we can sort of think about myoglobin as this subunit that's like extreme oxygen only if necessary. So it binds to oxygen with a higher affinity than hemoglobin, and it's like an oxygen reservoir in muscle tissue. So we would only really need it if that muscle was seriously needed with extra oxygen, and we had to release it. It is curve mediated, saturated mediated. So only when we reach a final low level of saturation in blood would we ever start breaking down that myoglobin to release the oxygen from it. If you think of other ways to process this, that means that it's non-circulating, exactly right, David, locked into muscles, and it's only necessary if you don't have much oxygen available. So where would we see, here's a kind of classic MCAT question, where would we see a higher amount of myoglobin concentration? In a bird like an emu, that's a bird that runs across the land, or a bird like a penguin that dives underwater? Where would we see a higher concentration of myoglobin? Yeah, definitely the penguin. exactly right. Jacob, Ahisa, Anna, because we're thinking diving mammal, something, or diving animals in general, it's going to require them to have higher oxygen reserves available for longer periods underwater. Nicely done. And again, that's a classic MCAT style question. We can tackle that by the breakdown. Now, the myoglobin curve, of course, as we see here, is hyperbolic. As Tyler points out, it quickly reaches that asymptote. The whole idea here is that it's reaching that asymptote at a very high affinity, so it's only releasing oxygen when we have that partial pressure dropping at the very, very final point. Now, this curve is not tested as frequently, unfortunately, as our sigmoidal binding curve, which we'll see in a second for hemoglobin, because really myoglobin only has that one oxygen, and it can only be released when we do have a super low partial pressure of oxygen in blood. Hemoglobin, though, is much more interesting for a lot of reasons. It absolutely has those 4 subunits. These are each of those heme binding areas. It does have the iron that is binding to oxygen in a simple redox reaction. And additionally, of course, as far as function, it's carrying oxygen in red blood cells, cooperative binding in general for each of those different pieces, which we'll see again in the overall binding curve in a second. And due to increased affinity, we've got the ability for it to bind more and more as each subsequent piece is changed from its conformation. There is one more point to make, and that's that this binding is based off of affinity. So we do have a pretty high affinity for things like oxygen, but unfortunately, iron has a higher affinity for some other things. Fortunately, it's not CO2. Fortunately, it doesn't have a higher affinity for CO2, but it has, unfortunately, a higher affinity for CO. So if you did have something like an increased percentage of carbon monoxide in the bloodstream, that carbon monoxide would be uptaken preferentially over oxygen. And it would lock up all of those positions, so we're no longer carrying oxygen around the system, we're carrying CO around the system. Luckily, CO2 is not as high in affinity, so oxygen preferentially beats that out, but it is something to keep in mind, that we could have things displacing oxygen and essentially destroying the reserve for the red blood cells. Now, as far as conformation goes, I quickly skipped over that a second ago because there's the original state, which you can call the tense state, where we don't have any oxygen bound whatsoever. We've released all of the oxygen. As soon as we have oxygen, we go through a transition state. And that transition state means that oxygen binds on, oxygen is our pink triangle for our purposes, and that changes the conformation in such a way that we have a conformational change in neighboring subunits. This increases a little bit the ability for oxygen to sneak in and attach to each of those. So as more bind and more bind and more bind, we have a much more relaxed state, which is much more able to bind subsequent oxygens. This binding curve process is easier to look at probably with a sigmoidal binding curve, which is this. Where if we think about it, that first oxygen is binding down here at the bottom at 0. The slope is kind of low. It's a little bit harder for that oxygen to bind on in the first place. The second oxygen now is up on that steep part of the curve. It's much easier to bind the second one. Not only do we have the conformation change, but we have 3 potential places for it to sit. Right? We only used up 1 so far. There's 3 more seats for it to jump in with. The third oxygen is going to bind still pretty aggressively, but it's leveling off a little bit, right? There's only 2 places for it to go left, and we don't need that oxygen to be bound as aggressively. Finally, the fourth oxygen should be a pretty flat line, should be plateauing out. There's only 1 more place for it to go. It's going to be easy to bind in there, but it's not as necessary for us to have that fast, immediate loading of oxygen. The other way to think about a binding curve is sort of in reverse, where we think about it loading from the lungs and working our way backward. So on the far right here, we can think about that as right there at the lungs. We have 100% saturation hemoglobin with oxygen, 100% partial pressure of O2 in millimeters of mercury. We're 100% ready to go. We're loaded. Now, that hemoglobin that's been loaded up with oxygen is good to go in general. The partial pressure, though, the bottom x-axis, is going to slowly drop as we go through active tissues. Even let's start with rest tissues, tissues at rest. If we have tissues at rest, hemoglobin traveling through the body is going to be dumping a bunch of that oxygen. So the partial pressure of oxygen is going to go down. Now, let's say it drops down about 60%, which is average for tissues at rest. Notice that 60% drop in partial pressure does not necessarily mean that the saturation on that hemoglobin has dropped significantly. In fact, the saturation on hemoglobin is only going to go down to about 75% because only about 25% of the bound oxygen is being used up by resting tissues. We still have about 75% of the bound hemoglobin oxygen ready to go to somewhere else. So let's suppose that we drop down a little bit more and we actually use active tissue, things that are really breaking down a lot more of that oxygen. Well, there's only a little bit more partial pressure left. So let's say we drop down to about 20% partial pressure of oxygen for active tissue. And this is, again, relatively standard. That 20% partial pressure of O2 means that there's a disparity here of about 60% or so of bound oxygen that just got thrown away, that just got used, that just got ripped off in order to be used by those metabolically active tissues. So the hemoglobin still has to oxygenate the rest of the body, which only takes about 25%, but the remaining 60 to 75% is going to be used up by those active tissues. Bonus question, remind me, what would happen if we dropped down even further than this? Let's suppose we're at like 10 partial pressure of O2, or 10 or 15 remaining on hemoglobin that could possibly be used. What's going to happen then when we've severely used up the hemoglobin load available? What's going to happen in that extreme case? Super active tissue. Yeah, that's when we go to myoglobin. Exactly right, Frank. Remember, myoglobin is that partial pressure saturation required breakdown. Only at that region do we go to myoglobin. Nicely done. Nicely done. So think about CrossFit, for example. What happens if you're in CrossFit and you're super overloading your muscles such that you need extra active tissue, you're breaking down all of the hemoglobin required oxygen. Now you're in anaerobic, lots of lactic acid, sure, Ahisha, sure, David, but additionally, using up a lot of myoglobin. And using up that extra myoglobin could cause some rupturing, could cause some rhabdo, for example, in very extreme cases where we actually have that myoglobin breaking down and traveling through the blood system because we have muscle fatigue or muscle break and muscle tissues that are breaking down, causing rhabdo, causing myoglobin release. Crazy stuff. Watch out when you're exercising. Now, going a little bit further here, as far as, sure, Tristan, sure, absolutely valid. As far as using that myoglobin or the hemoglobin binding curve, though, there's a couple more points I could make. First of all, as far as the curve in general, we really want to make sure that you know that there's a Bohr effect that could possibly take place if we also have a super high metabolically active release here. So tissues in active aerobic respiration are going to release lots of CO2. Exactly, Christopher. You should, just to be clear, know that CO2 buffer system. Like CO2 plus water gives us bicarbonate acid or bicarbonic acid, which, if we process that further throughout the body system, could give us hydrogen bicarbonate and extra acid. So yes, extra CO2 means that now we've got lactic acid as a byproduct, we've got extra hydrogen ions and extra CO2. So on the right-hand side, the net effect is we've changed the oxygen affinity by shifting it down and to the right. This down right shift is the Bohr effect. So that is due to the overall reduction in oxygen carrying capacity or binding capacity of hemoglobin. So Bohr effect, net overall effect based on that acidic environment. The flip side, though, is also true. Suppose we had somebody who instead is hyperventilating. You have a friend hyperventilating, you would say, gosh, okay, you're hyperventilating, you are rapidly decreasing the amount of CO2 left in the body. This is going to cause the reverse buffering system response based on Le Chatelier's principle, and then the concentration of hydrogen ions are going to make us increase the pH. With less CO2, we have more transfer, alkalosis, exactly right, Asia. So hyperventilating, we would tell somebody, hey, breathe into a paper bag, right? If you're hyperventilating, you're going into alkalosis, you've got a left shift because you want more and more ability to bind more of that oxygen, but you just don't have any oxygen available. So you breathe into a paper bag to recycle the CO2, bring the blood pH back down to levels that you would expect. Now, we often say paper bag, right? Because if they breathed into a plastic bag, then they're recycling CO2 for a while until they're only recycling CO2. And then we no longer have any oxygen available, and that could cause some other deleterious problems in a second. And then, yeah, exactly to Dell. Hyperventilation will be a left curve. Bonus question. When else, think about the entirety of human possibilities, when else would we ever see hemoglobin left shifting? I tried not to give you a hint there. Fetal hemoglobin, exactly right, Christopher. Nice, David. Absolutely. And again, as far as the MCAT is concerned, these are the big overarching pictures where we would expect that sort of process to happen. Nicely done. Nicely done. Now, at this point, as far as the overall session goes today, I really focused on the first two here, partly because I know that immunology comes up in some of the other channel sessions and other videos, and additionally because platelets, I'm going to hit you with 1 minute right now to talk about platelets overall, and I know you can always go back and take a look at the notes that we're going to put up for download in a second. So let's talk about the platelet requirement for test day, and I am going to pass right over immunoresponse because we will cover that in some other sessions. I really wanted to make sure you got overall questions about blood typing and overall binding curves, because sometimes we skip over that too fast in this session, I think, but that's where the MCAT spends the majority of their questions. Now, as far as the last piece here, again, this overall clotting disorders and problems you might have, we should know that overall cell fragments in bone marrow are producing and stimulating thrombopoietin, which is giving us production of our platelets and our megakaryocytes. And this overall production process in general is just to have a response, right? Thrombopoietin by the liver giving us, 3,000 or so platelets during every megakaryocyte lifetime, giving us 10 to the 11th platelets overall per day in the human body. This is massive, circulating through the system, but they're causing blood clotting only when necessary, right? The concept is we don't want to have any blood clotting unless we really needed that blood clotting. Here's the pathway, just to give you a whole bunch more information than you would ever need for test day. This is that massive cascade that would be required to have blood clotting, to have a reaction. Now, you don't have to know the vast majority of this, actually. Pretty much all the stuff at the top is useless for you for the MCAT. The bottom pieces are really the only ones that you need to know. But you should know that the convergence of these pathways is thrombin from prothrombin, which then promotes fibrin formation from fibrinogen, which then gives us that fibrous mesh. The other thing that you should know is that a breakdown of any part of this pathway in the zymogens causes an immediate problem in the rest of the pathway. So let's suppose that we had a breakdown in all of these zymogens that are floating around inactive in the blood, and we killed, let's say, this one, this 8 out of this entire process. Well, if we killed that one, then all of a sudden we no longer have that clotting factor requirement. For example, hemophilia A is a classic example of this. So the entire response mechanism is gone, and we would not have the thrombin and the fibrin clotting factors. That's the sort of way the MCAT would test us on this. Or with a response mechanism. Suppose that we had hemophilia A, then we would have to have an infusion with the missing factor, or a blood transfusion in general, or a gene therapy to fix that, something like that. And again, the classic MCAT question would be something like this. Suppose we had a process where we were missing those zymogens, or we had extra zymogens, what would we see as a response? Now, to Frank's question, it could be either one of these starting pathways at the top, but the bottom piece is the only stuff you really have to know for the MCAT. That's the common pathway, after the intrinsic pathway or the extrinsic factors. Good question. At this point, though, I know that we are right here at time, and I really hope that you appreciated the extra time we spent on the blood typing and also the curves so that we didn't just rush through those. And if you have any remaining questions, I am going to be on